This is National 5 Biology Unit 2.4 Variation and Inheritance. So up to this point, in terms of DNA, we've looked at from Unit 1. You've had the shape of DNA and the bases and so on. And you'll know that we have chromosomes and they contain genes. And we're just kind of taking that forward a little bit and looking at some more genetic terms and how different people have different characteristics. So to begin with, first session, state the meanings of the following words, so gene and phenotype and allele, and then we're going to look at two types of variation, and they are discrete and continuous. So the first genetic terms, and these are the sort of things maybe we'll do as we go through this. We'll try and get them into our heads, but this would certainly be one where flashcards would be a good idea to try and get them into your head. So a gene is defined as a section of, that is, chromosome, and the gene is made of DNA. So it has a DNA sequence which codes for a protein. Okay, so remember that from Unit 1. So the DNA provides the code to make mRNA. The mRNA then goes to the ribosome where the protein is made. Okay, and then the protein, once produced, controls a something, for example, the gene for eye colour. So eye colour would be an example of a characteristic. So you might know that word already, Gene. This one I think should be new. So phenotype, so that is the physical characteristics, and that begins with pH, which might help. Remember, so the physical characteristics of an organism. So if you see someone with brown eyes, you can say that their phenotype for eye colour is brown eyes. Now, the second term, or the third term, sorry, is allele. So they are the different versions of a particular gene. So you look at someone and you say that they have the phenotype for brown eyes. The allele is the different versions of a gene. So they produce different characteristics, different phenotypes. So you have got the eye colour gene, but then because you have got different eye colours, you have different alleles, so different types of gene which produce different phenotypes. So you would talk about the brown and blue alleles for the eye colour gene. For hair, you could have the straight-haired allele, the curly-haired allele, for example. And variation is simply the differences between individuals. So if we look at these individuals here, you can see we have differences in skin colour, we have differences in height as well, are the two most obvious ones. Those are quite odd looking shoes, aren't they? I think they're sandals. Yeah, so variation is just simply differences between individuals within a species. And the reason for variation is because you have got the genes of two parents which are combined. Okay, so you'll have some characteristics which will come from your mother's genetics, and you'll have some characteristics which come from your father's genetics. So have a look at this on YouTube. This is the name of the video, so just search for that, and that will give you some more examples and talk you through a little bit more about variation. So just to give you some examples of genes, we sometimes think that, I guess it's easy, a human lifetime is 80 to 100 years, and you might think that things are relatively stable, but things change over time, even in human genetics. And blue eyes are a relatively recent mutation. The other one, which has been, I don't know if discovered is the correct word, but knowledge of it has come to the fore, is the MC1R gene. And if you live in Scotland, then you'll be aware of this, because instead of calling it the MC1R gene, it's called the ginger gene. So most places around the world, this particular type of variation is not present. It's kind of limited to a smaller number of countries. However, the origin of it has been traced back to Asia 60,000 years ago. So one person had this mutation which resulted in red hair colouring. But it's not just the hair colouring, is it? So you've got red-headed, but also the individuals who have got red hair are paler skinned, and during the summer this is not great. However, going back in time, go back 60,000 years ago, your average person maybe died before the age of 40. So mutations like this could be helpful. So if you were living in Northern Europe where there was low sunlight for 7 months of the year, then if you have got that pale skin, then it's easier for you to get vitamin D, um, which originates from sunlight, or sunlight allows it to be produced. So actually, this is an advantage. And when times were tough, this could have been the difference between life and death. So this was an advantageous mutation. And worth mentioning as well that there's no evidence for effects on personality. So all of that stuff about fiery redheads and so on, I don't know if it's no evidence to back that up. So we've got two types of variation, and this is the first one exemplified by this girl here. So this is called discrete variation. So discrete variation, as a definition, is when a phenotype is controlled by a single gene. Okay, and the measurements fall into distinct groups. So, for example, the ability to roll your tongue, it's either a yes or a no. You can either do it or you can't. There's no range of tongue rolling. Another one here, which is a good example, is blood groups. So for blood groups, you've got a limited number of types. Okay, so you've got 4 there. So discrete variation, it's either one or the other, or in a small number of groups. So you can see you've got the percentage of the population there. And because discrete variation is something which falls into a small number of groups and is not a range, it means that you can plot it as a bar graph. So if you see a graph of data and you were asked which type of variation it is, if it's a bar graph, then the chances are it's going to be discrete. The second type is continuous, and exemplified here by the height of these celebrity males, from 5 foot, Danny DeVito, all the way up to 5'10", Johnny Depp and George Clooney. So continuous variation is when phenotypes are controlled by more than one gene, and this can be, you can use this word to describe that, and that's polygenic. Okay, so polygenic means controlled by more than one gene, and because the phenotype is controlled by more than one gene, you get a wide range of values. So you have a minimum and a maximum. So the example that I'm giving you here is height. Okay, and here it is here. So as I kind of mentioned when I was talking about discrete variation, for continuous you can plot it as a line graph. So we've got the bar or the histogram underneath, but you can see you get a nice what's called a bell-shaped curve. So the majority of people are in the range within the middle, but you do have some at the extreme minimum and extreme maximum. So continuous variation, usually drawn with a line graph, and it's controlled by more than one gene, polygenic. So can you draw that table, please, and press pause. And now I would like you to copy those into the correct part of the table for either discrete or continuous. Once you've done that, press play again and I'll tell you if you're correct. Okay, done that. This is what you should have. So you should have 4 in each side. So for discrete variation, you would have control by a single gene, displayed as a bar graph. Measurements fall into distinct groups. An example would be tongue rolling ability. And then for continuous variation, control by more than one gene, so it's polygenic, displayed as a line graph, shows a wide range of values, and finally, an example would be height. Okay, have a break now, come back another day. Okay, here's a starter. So from last time, we were looking at continuous variation and discrete variation, looked at differences between them, and put down numbers 1 to 8, and I just want you to write beside it CV if you think it's continuous variation, and DV if you think it's discrete variation. Press pause and do that. So number 1, blood type is discrete. Weight is continuous, that's a range. Height 3, that's a range, that's continuous. Earlobe attachment, it's either attached or it isn't, so that's discrete variation. Tongue rolling 5 is discrete. 6, heart rate, that is a range, so that's continuous. The length of a leaf, that is continuous, so that is 7 is CV. And 8, eye colour, that is discrete because there are a limited number of eye colours. So the next section of this, we're going to take it on a bit further, and we're going to bring in some more words and relate it to inheritance. So we've got genotype, dominant, recessive, homozygous, and heterozygous. So we talked about alleles before, and remember an allele is, or alleles are the different versions of a gene. Now, if you've got an allele which is dominant, that means that it always shows. So even if you've only got one copy of it, so you get an allele from your mother, on the chromosome you get from your mother, you get an allele from your father on his chromosome, and you only if it's a dominant allele, you only need one copy of it. So if you get it from your mum but not your dad, it's going to show. You're going to have brown eyes. And then recessive is the opposite. So recessive is an allele which only shows when there are two copies of it. Okay, so dominant, you just need one copy of the allele to have the phenotype, to have the characteristic. Recessive, you need two copies. Okay, next terms, so genotype. So the genotype shows an individual's alleles, okay, for a particular characteristic. And usually, if you're asked for a genotype, then you would write it as letters, and it's either capital letter or a lowercase. And related to genotype, heterozygous is when the two alleles of a particular gene are different. So you can see from this one, you've got an uppercase and a lowercase. So you've got two different alleles, and that is what you would call a heterozygous genotype for that characteristic. And then homozygous, that is when the two alleles of a particular gene are the same. So you can see here, you've got capital letter, or the other possibility, lowercase, lowercase. And you might see this term sometimes. This is called a true breeding genotype if it's homozygous. Okay, so given that information, I want you to try and copy and complete that. So fill in the table and use the words that I just described to you. So that was homozygous, heterozygous, recessive, dominant, phenotype, genotype. Try and put them in the correct place. Okay, press pause and do that. So what you should have is the headings for the table. These are the letters of that show the two different alleles. So that is genotype. And then this is what the person actually possesses, what you see. So that is the phenotype. This person here with brown eyes is big B, big B. So that means that they are homozygous for that big B allele. This one here, small b, so they're also homozygous, but it's just for this other allele. And then this last person, big B, small b, so that means they are heterozygous. And down here, big B, brown eyes is the dominant allele. And we know that because this information here. So if you only have one copy of it, then you express the characteristic, you have brown eyes. So that shows that it's dominant. Whereas small b, blue eyes, it's recessive because the characteristic is only apparent if the person has both alleles. Okay. So try this then. Okay, again, press pause 1 to 8 and put these in the correct place. So number 1, gene, that is A. 2, allele, is D. 3, phenotype, the physical characteristics, that's G. 4, genotype, the kind of giveaway here is usually written as letters, so that's E. An individual's alleles, usually written as letters. Dominant, allele which shows if there's only one copy of it. Recessive, an allele which only shows when there are two copies of it. And then heterozygous, 7 is B when the two alleles are different, and 8, homozygous, is H when the two alleles are the same. Okay, have another break. Okay, here's a bit of revision again. I don't know why I've messed up. This should say numbers 1 to whatever. It's 9 to 16, but anyway, press pause and see if you can write down what each of these terms means or give an example. So the first one, the gene, you've got the kind of, you're working from what's down here. So the gene here is for eye colour. And then allele, the definition is the different versions of a particular gene, and they produce different phenotypes. And then phenotype 11, the clue here is, tells you the example is brown eyes. The phenotype is the physical characteristics. And then genotype, the individual's alleles for a particular characteristic, usually written as letters. So for 12, you could just have anything there. Well, big B, big B, big B, small b, small b, small b would be okay, but I suppose even if you had, you get two letters there, it's correct. Dominant is an allele which always shows, even if there's only one copy of it. So the example here is brown eyes, big B. An allele which only shows when there are two copies of it, so 14 would be blue eyes allele, which is small b. And then homozygous, well, I guess you can use heterozygous to help you here. Heterozygous is when two alleles of a particular gene are different, so that means homozygous is when the two alleles of a particular gene are the same. And you might have said that this is also called true breeding. And then finally, an example of heterozygous. Well, if an example for homozygous is big B, or small b, then heterozygous would be big B, small b. So we're going to get into looking at now if we've got the parents' genotypes, what the offspring looks like and what the generation below that would look like, and you do that using Punnett squares. So this is Larry Cotter and the recessive allele. How are wizards made? So here's the information. Being a wizard or a muggle is all decided by genetics, and all humans, including wizards, receive one allele from each parent. So the allele to be a wizard is small m. So that means wizards need to be homozygous recessive, small m, small m. Muggleness is big M, so it's dominant. And that means that if you're a muggle, then your genotype must be big M, small m, or big M, big M. Yep, there you go. Big M is dominant, so you can only be a wizard if you've got no big M allele. So we're going to go through this, and I think there's about numbers 1 to 8-ish. So write down the answer for that. So easy one, isn't it? So if they're both small m, then Colin must also be small m, small m. Number 2, the Cotters. It's the same again, isn't it? Small m, small m. So we started off with wizards. Okay, and same with the Weasleys. They all have small m, small m. Agnes is small m, small m. So again, her parents, what's 3 going to be? They must be big M, small m, because it says here that her parents are muggles. So here's where it's got a bit, this is where it gets a little bit more complicated, isn't it? Because she could have got the big M from them, but she didn't. Just by chance, she got small m, small m. So despite her parents being muggles, because they contained the wizard alleles, she could become a wizard. Similarly, Lafferty McLafferty, so his mother was a wizard and his father was a muggle, big M, small m, which means that his father must have had the alleles big M, small m, so that he could give him the other small m allele. Okay, so if you're looking from the parents before they had the child, if the mother was a wizard and the father was a muggle, then they must have known there was, their child could have been one or the other. So if they have children, 4, they would be small m, small m. And this is the way that you do it. So we'll start off with an easy one. So this, we talked at the beginning of the learning intentions about Punnett squares. This is a Punnett square. So what you do is you just do that times that and put the 2 letters in there and repeat for the other 3. So draw that out and write it in just as it gets slightly more complicated than this. But good to write it out just to get into the way of doing it. So that's what it should look like. So all of their children are going to be wizards. Here we have Liz and Rod. They're going to have children. She is a wizard, he is a muggle. So do the Punnett square for that one. Press pause and do that. So what you should have is something that looks like this. It doesn't really matter if you have the lowercase first, but it's generally you do the capital first. So what you've got here is Liz is a wizard, but because Rod is homozygous dominant, it means that all of their children are going to be heterozygous, so they're all going to be muggles, despite the fact their mother is a wizard. Okay, try this one. So here we've got Versace and Frederick. Okay, do the Punnett square for these two. So for this, I hope you've done it. Number 7, you should have that first of all, and then that gives you this. So you're going to have 2 wizards and 2 muggles. Okay, and then I'll let you do this by yourself. So given that Agnes's parents, Mr. and Mrs. Granger, are heterozygous, what would you expect their 4 children to be? So press pause and do the Punnett square and then write down how many muggles, how many wizards. Okay, do that now. So what you should have for this, your initial Punnett square should look something like that. Again, if you've got the capital letters the other way around, it doesn't matter. So what you would have is that. So you have 3 muggles and 1 wizard. So this is one of the sort of quirks of genetics. So you've got 2 parents who do not have the wizard characteristic, but they carry the gene for it, which means that one of it can skip a it's basically almost skipped a generation, hasn't it? But possibly. So you've got the wizard characteristic reappearing in the next generation. So some of you can have a think about this in your family. If both of your parents have brown eyes, then potentially you or one of your siblings could still have blue eyes if they both carry that gene. So, a few more genetic terms just to familiarise yourself with before we have a look again at Punnett squares. So P1 is the parental generation. So think back again to the wizards and you had your parental generation, and then the generation below that is called the F1. And it is the first filial generation, and that is all the offspring of the P1 generation. And then finally, you have the F2 generation, and that is all the offspring of the F1 generation. So I suppose if you think about it as your family, you could almost think of P being, your grandparents, and then F1 your parents, and then F2 would be you. Okay, or P being your parents, F being you, and then F2 being your future children. And what we have here is what's called a monohybrid cross. So this is what we've been looking at with our wizards and muggles. So all we were looking at with the wizards and muggles was whether or not the person was a wizard or a muggle. So that's one characteristic. So we've got P's here. The reason why is because Gregor Mendel, I think he was a monk, and he was one of the early investigators, and he found out he did crosses with P's, which helped work out what was going on in terms of alleles and what was passed on. So monohybrid cross, the study of one characteristic, and those terms we had before. So there's are shown here. So there's P1, there's F1, and there's F2. And again, because of the genotypes here, we have a generation that's all round, but then wrinkled appears in the F2 generation. Okay, so see if you can copy and complete that. So you're going to have to do underneath Punnett squares, I think. So you certainly need to do one for the F2, maybe not for the F1, but press pause and see if you can complete that. So we've got two parents who have got curly hair and straight hair. They're both homozygous, dominant and recessive, and work through what the next two generations their genetics will be. So what we've got for alleles is going to be big H times small h, and as a result of that, the whole F1 generation are going to be curly haired. They're all going to be big H, small h. Then to do the F2 generation, you need to draw out your Punnett square. So your Punnett square is going to look like so, and you fill it in and you get that, which means that the F2 generation, you're going to have 3 with curly hair, because they only need one copy of big H because it's dominant, and then you're going to have 1 with straight hair. So that's your monohybrid cross. So the Punnett square, you use that to work out, generally if you see it in exam papers, it's used to work out the F2 ratio. And something that's worth pointing out, we had that with the curly hair, we had a 3-to-1 ratio. However, it's worth mentioning that with the Punnett square, it's a predicted ratio, and you might not necessarily always get the 3-to-1. And the reason why is because fertilization is a random process. We've got millions of sperm with different, it's a kind of 50-50 in terms of what's going to happen. So it's a random process. And also, if you've only got, say, 4 offspring, then you've got a small sample size. Okay, so that's just sometimes you might get asked why, if there was an experiment and it wasn't the 3-to-1 ratio that you expected, why might that be the case? And this is why. Okay, time for a short break, and then we'll conclude this by just doing a wee bit of revision. So to begin with, there's 1 to 5, and again we'll use our eye colour as an example. So I want you to write down the answers for that. Press pause. So number 1, the gene here is for eye colour. But they've given us the alleles and the phenotype. So the possible genotypes, there are 3 possibilities: big B, big B, big B, small b, or small b, small b. Then the dominant heterozygous genotype is going to be that. Don't really need the word dominant there, I suppose. The dominant homozygous genotype is going to be big B, big B. And then the recessive genotype is going to be small b, small b. So we'll have a look at a couple of past paper questions now. So press pause and do this. So this is from the 2016 paper. Have a read through, write the answers, and then press play again. So the following diagram represents part of a family tree showing the inheritance of hitchhiker's thumb, where the thumb can bend back as shown below. So we've got that first generation, P1, F1, and then F2, and it's telling us that straight thumb is black and hitchhiker's thumb is white. And it says complete the table below for individuals A and C. So if you go down here, it's telling us that big T or big T small t is straight thumb. So that must mean that straight thumb is dominant. Okay, so small t then must be the white one. So that's hitchhiker's thumb. I'm not going to do all of these because I struggle to write with this. But that would be big T. That would be small t, and then straight thumb must be big T, or big T, small t. And actually, once you have got that information, a good thing to do is just to go back and write them all in. So you can now actually go back and put, okay, that's small t, so is that, so is that, so is that. And if you've got that information, then you can go back and work out everything else. So let's have a look at this then. So we're looking at individual A, which is here. Now, this individual has got a straight thumb, so that means they must have big T. Okay, they just must, because that's telling you there that it's dominant. Okay, so the second allele, is it going to be big T or small t? Now, if it was big T, then it would have to give big T to all of its children. However, you can see that two of them are small t, okay? So that must mean that its second allele must be small t. So that's A, big T, small t. If all of these were black, if they were all straight thumb, then we could say that one was big T, but because they have got, you've got your recessive ones in here, they must have that recessive allele. And then the second one, individual C, who is down here, and they are hitchhiker's thumb and small t, small t. Okay. B, in a survey of 90 students, it was found that 25 of them had hitchhiker's thumb. Calculate the number of students with straight thumb to hitchhiker's thumb as a simple whole number ratio. So you're going from straight thumb to hitchhiker's thumb. So that means that if 25 had hitchhiker's thumb and there were 90 in total, then 65 must have had straight thumb. 65 plus 25 gives 90. Okay, so it is 65 to 25, and then to simplify that, you divide both sides. So 65 and hitchhiker's thumb would be 25. Okay, so to simplify that, you get 13 to 5. Okay. And then part two, the predicted ratio was 3 straight thumb to 1 hitchhiker's thumb. Why was the predicted ratio different? So we mentioned this. Two things you can. Here's another one. So press pause and I'll go to this. This is from 2018. So for this one, tongue rolling is an inherited characteristic controlled by different forms of a gene. Big T roller represents the dominant form of the gene, and small t non-roller represents the recessive form. The family tree diagram shows a pattern of inheritance of the characteristic. So they've put in white colour is tongue roller and the dark colour is non-tongue roller, and it tells you that non-roller is the recessive form. So this here is going to be small t, okay, which means that this one must be, if it's a tongue roller, then that is dominant. So if it's got the white genotype, it must be either big T, the heterozygous one, or big T, okay. So first one, use letters from the diagram to identify all the individuals in the F2 generation. So the generations, this one is going to be P1, and then below that is F1, and then below that is F2. So F2, G, H, I, J, K. Part 2, give the genotypes of individuals E and F. So E has got the darker colour, so that means it's recessive. So E must be small t, small t. Then you've got F. So F is over here, and F has got is a tongue roller, which means that it must be one of these. So it's definitely got T. Okay, and remember, just genotype is when they ask you for the letters. So it must have a T. So the second allele must either be big T or small t. So to find out, we look at the children. So if we look at these children, that one has got a big T, definitely. That one's got a big T. But if you look at K, look, K is a non-tongue roller. So that means that K must be small t, small t. Now we look at the parents, definitely get a T from this one, but it must have definitely got a small t from E, but it must have got a small t from F as well. Okay, so that means that F is going to be big T, small t. Then part 3, complete the Punnett square to show the gametes and expected genotypes of the offspring of E and F. So just that leads in from the last question. So E must be small t, F must be big T, small t. So you just fill that in like so, and you get 2 marks for that. I guess the 2 marks, you get 1 mark for doing the parental alleles here, and then you get a mark for filling in the Punnett square. And then finally, state the type of variation shown by tongue rolling. So remember, you've got 2 choices, either discrete or continuous. Discrete, there's a limited number of forms of it. It's not a range, and this is what we're seeing here. So B is discrete.